Recording has begun.
Okay. Well, good morning and welcome to the High Fidelity Seminar Series. It's my great pleasure to introduce our High Fidelity Seminar speaker this morning, Dr. Hillary Andrews. And Dr. Andrews serves as the Director of Regulatory and Research Partnerships at Friends of Cancer Research, or FRIENDS, where she supports the development and implementation of the organization's research and policy agenda. Dr. Andrews collaborates with diverse teams to identify challenges in cancer research and create solutions that improve and accelerate cancer care for patients. Prior to joining Friends, Dr. Andrews worked as a consultant at Avalier Health, where she supported clients in navigating the regulatory strategy and FDA policy environment to improve market access. And Dr. Andrews works with Teresa's Research Foundation to improve cancer research through establishing relationships between cancer researchers and patient advocates. Dr. Andrews completed a postdoctoral fellowship in tumor biology at Georgetown University's Lombardi Comprehensive Cancer Center. She earned her Ph.D. in endocrinology and animal biosciences from Rutgers University and her BS in behavioral neuroscience from Lehigh University. Today, she will tell us about our efforts to advance the use of novel technologies through collaboration. Although we're virtually together, please welcome Dr. Andrews.
Thank you so much for the introduction. I'm really excited to be here to share a little bit about some of the work that's ongoing at Friends of Cancer Research. Friends is a nonprofit based in DC with the mission to drive collaboration among partners from every healthcare sector to power advances in science, policy, and regulation that speed life-saving treatments to patients. We accomplish this mission largely through our research partnerships and policy and regulatory initiatives. Today, I'll be focusing on two of our research partnerships, but I wanted to give you a little bit of background about all three of the key areas that we have. The first is supporting early endpoint development. For this aspect of our projects, we ask the question, can ctDNA serve as a predictive early endpoint in drug development? As an approach, we perform aggregate analyses to support the use of ctDNA as an early endpoint. And for this, we have a project called the CT Monitor Project. Our diagnostic policy to improve consistency efforts ask the question, how do assays align and can we find consensus to improve concordance? Our approach here is to provide a common data set to diagnostic developers and assess concordance. And we have a few projects that fall under our diagnostics harmonization projects. And finally, some of the work I won't be reviewing today, but just wanted to make you aware of is our real-world evidence for regulatory decision-making. Here we're asking the question, what is the best approach for evaluating real-world data to identify drugs that benefit patients? For the approach, we align on a common data plan to assess whether real-world data can inform clinical outcomes. And here we have a few projects in our real-world evidence portfolio. First, I wanted to walk you through our CT Monitor project. The CT Monitor project asks the questions, is ctDNA a predictive early endpoint in drug development? Again, our approach here is to perform aggregate analyses of ctDNA data to assess relationships between ctDNA levels and long-term outcomes. Our hope that this impact will ultimately lead to in the long term using ctDNA as an early endpoint to get effective therapies to patients faster. This project is ongoing and began in 2018 at our Friends annual meeting where we had a discussion about how to align more effectively on using ctDNA and established a white paper. It was the beginning of the project. We launched CT Monitor in 2019 as a pilot project in non-small cell lung cancer with patients who were treated with an anti-PD-L1. The goals here were feasibility, aligning on methodology, and it was our first attempt at looking at whether changes associate with long-term outcomes. We've subsequently established CT Monitor Step 2, which is a larger project with 3 modules in multiple cancer types. The goals here were expanding to more cancer types and treatments and informing ongoing development and assessment in the field. In addition, in the background, we've been working on a roadmap, which we presented about in 2022. And the roadmap is sets the stage for how to ultimately use ctDNA as an early endpoint long term. We also had a project where we looked at baseline levels of ctDNA across multiple diagnostic developers, and we have conversations with FDA that are ongoing. For CT Monitor Step 1, our project includes a collaborative group of representatives from pharmaceutical companies, diagnostic companies, FDA, and academia. CRAB, or Cancer Research and Biostatistics, serves as our independent statistical analysis team and data aggregator. And Naveen Zarifa from the NMD group serves as our statistical consultant. For our objectives, we wanted to investigate the feasibility of harmonizing ctDNA data measured from different assays using different collection schedules, to align on a methodology to combine clinical data from multiple trials in lung cancer, and characterize associations between ctDNA values and tumor response. Our question is whether trends that are observed in smaller independent data sets can be replicated in a larger combined data set. For our Step 1 data set, we set out to identify clinical trials that had available data, so it was already published and previously collected. Patients were diagnosed with advanced non-small cell lung cancer. They were treated with an anti-PD-L1 therapy. They had to have tumor response evaluation and overall survival and progression-free survival data. And they had a ctDNA measurement using variant allele frequency, or VAF, at baseline, plus one or more follow-up samples on treatment. And the figure on the right just shows the 5 studies that we identified for our Step 1 data set and the timing to demonstrate that there were different times when ctDNA was collected in each of the various studies. We had a few challenges that we needed to overcome because we were combining such a heterogeneous population of patients. For the first challenge of overcoming the heterogeneity of the population, we used stratified statistical models with a different baseline hazard or risk for each cohort. We had varying levels of clinical detail that were provided, and so we created derived variables by collapsing data into binary and categorical variables to maximize inclusion. We had inconsistent timing of ctDNA sampling, so we applied filtering requirements and defined baseline as ctDNA that was collected 14 days before, up to 14 days before treatment start, and ctDNA on treatment, or T1, as no later than 70 days from treatment start. And finally, we had different assays that were used to measure ctDNA. And to overcome this, we calculated a change in ctDNA levels to compare ctDNA metrics across assays rather than using an absolute change. For this analysis, we used a 50% decrease in variant allele frequency, or VAF, as our cutoff to define molecular response. And we see a clear separation on the Kaplan-Meier curve when we're looking at those patients who have a strong decrease versus those who have an increase or weak decrease in their overall survival. So we see consistent and robust associations between reductions in ctDNA and long-term outcomes. In this case, I'm showing you long-term outcomes of overall survival, but we also saw similar results when we looked at progression-free survival. We see that disparate ctDNA studies can be harmonized using various statistical methods and other approaches, and that the strength of association remains after accounting for clinical covariates. So once we finished our Step 1 analysis of a smaller cohort of 5 clinical trials with 200 patients, we moved on to CT Monitor Step 2, which includes 22 clinical trials, over 3,000 patients, 8 different advanced tumor types, and 16 different therapies. We took this data set and divided it into 3 modules. The first was assessing advanced non-small cell lung cancer treated with tyrosine kinase inhibitors. The second is Module 2, advanced non-small cell lung cancer treated with an anti-PD-L1 or chemotherapy. And our Module 3 is our advanced solid tumor, which is treated with either an anti-PD-L1 or TKI. We kind of call it our catch-all, so it's multiple cancer types and multiple treatment modalities. The next few slides, I'll walk through some of our findings from Module 1. The Module 1 analysis included 8 clinical trials with 8 different tyrosine kinase inhibitors. We had 1,015 patients, and there were 5 different assays to assess ctDNA. We harmonized the data across different time points in addition to harmonizing across different assays. The index date for the analysis was treatment initiation or randomization. We defined baseline ctDNA as up to 14 days before index and our on-treatment ctDNA as up to 10 weeks from the index. Across all trials, the cadence of ctDNA and first RECIST assessment varied. And so I'm just showing you here the various time points across the different assays of when we had ctDNA measurements and when the RECIST timing was occurring, the first RECIST, and that was included as either T1 or the first RECIST for the multiple time points.
Dr. Andrews, you probably know that we lost your slide, right?
Oh, no, I didn't. Here, let's see if I can get you back. Sorry about that.
Otherwise, we can help.
Should be able to. Is that good?
Perfect.
Sorry about that. Okay. Thank you for letting me know. So for our approach for Module 1, we had a few separate questions that we wanted to ask. The first was aligning with our overall project goals. Do early changes in ctDNA level associate with long-term clinical outcomes? Here we performed Kaplan-Meier plots and multivariable Cox proportional hazards models that were stratified by cohort and included demographic and clinical confounders. We asked whether changes in ctDNA levels complement first RECIST, and then whether combining changes in ctDNA levels with first RECIST improved associations with outcomes. So for research objective 1, or our first question, whether changes in ctDNA associate with long-term outcomes, we first defined the change in ctDNA metrics. Here we were using MaxVAF, and we calculated change in ctDNA similar to what we did in step 1, where we looked at a change from T0 to T1. For those with multiple ctDNA values within the first week, we use the best or the lowest ctDNA for T1. We created molecular response categories based off of a 50% decrease, a greater than 50% decrease, a greater than 20% increase, and an intermediate category, which was a 50% decrease to a 20% increase. We also noted that there were a lot of patients who had non-detected ctDNA, either at baseline or on treatment, and so we created an ND category for those who had non-detected ctDNA at both categories. When we were categorizing the samples by percent change, we did not see a separation of the Kaplan-Meier curves. And patients with detected baseline ctDNA who went on to have non-detected ctDNA on treatment were included in the decrease category. And we determined that categorizing these samples by percent change did not demonstrate separation. And so we moved on to create categories based on detection, because you'll note that there are quite a few patients that are in the decrease category. We ultimately had 41 patients in the intermediate and only 17 in the increase category. So we created categories based off detection. So whether you're non-detected at both T1 and T0 and T1, detected at T0 and then became non-detected, so a clearance category, or detected at both, regardless of whether there was an increase or decrease. And here we see that patients with detected ctDNA at both baseline and on treatment had the worst associations with overall survival, regardless of whether their ctDNA increased or decreased. We performed a cut point analysis of this group, and we were unable to identify a cut point that separates the data. And so patients with our main takeaway is that patients with non-detected ctDNA on treatment have stronger associations with improved survival compared with patients who have detected ctDNA levels on treatment. We decided to use these ctDNA categories moving forward to define molecular response. Once we had our ctDNA categories, we decided to compare molecular and radiographic response. We combined the CR and the PR, so the complete responders and the partial responders, into a responders category. These are based off of RECIST for radiographic response. And we combined the SD and the PD into non-responders. And we see that the distribution of responders versus non-responders across the different molecular response categories for first RECIST and for BOR is similar, and that we don't really see differences between the different ctDNA categories. So subsequent focus studies that I'll be walking through focus on first RECIST rather than the confirmed or best overall response. And that's for two reasons. The first is that the ctDNA on treatment time point is within 10 weeks, and so is the first RECIST. Also, the BOR timing, or the best overall response timing, was variable and sometimes took many months, which meant that indexing our analysis would be challenging for our Cox modeling. When combining ctDNA and radiographic response, we performed 4 different analyses. The first was to look at radiographic response alone with no ctDNA included. So just looking at responders versus non-responders and associations with outcomes. The next was to look at radiographic response with changes in ctDNA outcomes. We then compared the models to see whether combining molecular and radiographic response improved associations with outcomes. And then finally, we looked just within the PR and the SD categories and whether ctDNA was able to associate with long-term outcomes within those categories. So for our first analysis, we see that there is no apparent separation of the curves between responders and non-responders at first RECIST and their associations with overall survival. For analysis 2, we created 6 categories combining molecular and radiographic response. So you can see that non-detected responders are in dark blue, and then the non-detected non-responders are in light blue. And we see that there's a bit of overlap in the non-detected. So whether you're a responder or not does not separate out the curves. For the dark gray and light gray lines, which are detected who become non-detected responders versus non-responders, we don't see much of a separation. And for the red category, or detected and detected, while there are differences there, we see that the molecular response categories continue to drive associations with long-term outcomes. Overall, we felt that incorporating radiographic response at first RECIST into molecular response categories does not improve the degree of association with survival for those with non-detected ctDNA on treatment. And so for analysis 3, we asked whether combining molecular and radiographic response improved associations with outcomes. We compared our full model to a reduced model, and here the full model includes both changes in ctDNA and first RECIST with our covariates, and the reduced model looks at changes in ctDNA only with covariates. And we performed a likelihood ratio test for association between overall survival and first RECIST above and beyond changes in ctDNA, and overall see that radiographic response at first RECIST does not provide additional value in characterizing the association with overall survival beyond the contribution of molecular response. And so we moved on to look within just the categories for stable disease and for our partial responder categories. And we see similar patterns to when we look at the overall data set. So for patients who were classified specifically as stable disease at first RECIST, molecular response is able to differentiate associations with overall survival. We think this is really important for the field because in addition to using just ctDNA, when using ctDNA within the stable disease category, we're able to get more granular information about how patients ultimately fare on treatment. And so our key takeaways from Module 1 are non-small cell lung cancer treated with TKI analysis. Harmonizing data across the 8 clinical trials was feasible. In advanced non-small cell lung cancer treated with tyrosine kinase inhibitor, among patients who had detected ctDNA at baseline, patients with non-detected ctDNA on treatment were associated with improved overall survival and progression-free survival. I didn't share those data, but we saw similar patterns compared to patients with detected levels of ctDNA on treatment. Molecular response associated with overall survival and progression-free survival, while radiographic response at first RECIST did not. And among patients who were categorized as having stable disease at first RECIST, at first RECIST, molecular response identified patients with improved associations with overall survival and progression-free survival. So some of our strengths and limitations of this analysis. It was a large data set of 8 clinical trials with 1,015 patients and 8 distinct TKI regimens. We saw clear and unifying results across a wide range of features. However, we did not have an external validation data set. There was variability among the assay types and sampling timing for ctDNA and RECIST. And so we were able to perform our analyses stratified by cohort, which strengthened our confidence in the results. We used various harmonization approaches, and we're still able to see that the results came out as we had anticipated. However, there's potential that the differential in features is due to variability despite our sensitivity analyses. We had patient-level data, so we were able to perform multivariable models that account for patient-level confounders, and we interrogated ctDNA metrics. However, we were unable to answer some questions because the data were anonymized. And these were retrospective analyses. So it provided us with an opportunity to analyze multiple data sets with different approaches to data collection. However, a prospective analysis would be pre-specified and allow design elements to be harmonized. So contextualizing our findings so far, we know that Module 1 is not necessarily a validation of Step 1. We have different treatments, TKI versus PD-L1. We saw different methods of defining change, so detection versus a percent change. However, in both cases, we saw that molecular response associates with improved outcomes. We're looking forward to our Module 2 analyses, which focus on advanced non-small cell lung cancer, again with an anti-PD-L1, but now we'll also have a chemotherapy arm. So we'll be able to further investigate results from Step 1 and consider a different treatment regimen. And so for Module 2, we're in the process of analyzing these data, but just wanted to provide you with an overview of the analysis and some of our plans. We're again in advanced non-small cell lung cancer. We have 4 randomized controlled trials of anti-PD-L1 versus chemotherapy. The assays are tumor-informed or PBMC cleared, which is not something that we had previously. So this helps to remove CHIP, so unwanted variants that come from the blood, and helps us to ensure that we're looking at tumor-derived variants rather than either CHIP or any genomic alterations the patient may have from their genetics. We also looked at the majority of patients have 2 on-treatment ctDNA time points, and this is just a quick little sampling diagram to show you that we're looking at multiple groups that have more than one ctDNA time point, which will allow us to perform some time point analyses. So our research objectives for Module 2, the first, again, are changes in ctDNA levels associated with long-term outcomes in patients treated with an anti-PD-L1. Research objective 2, what are the dynamics of ctDNA levels over time, and what is the interplay with these and long-term outcomes in patients treated with anti-PD-L1? Included within this question, we'll ask about things like whether a confirmatory time point similar to RECIST is required to define molecular response. And finally, for research objective 3, whether changes in ctDNA levels associate with long-term outcomes in patients treated with chemotherapy, and how does this compare with those who are treated with an anti-PD-L1? Some of our outstanding questions that we hope to answer with our Module 2 analysis. We know that there are differences in assays, the limits of detection when you're defining response based off of a detection. We had a lot of conversations with our working groups about the limits of detection of the different assays. What's the level of alignment that will be necessary for assays moving forward if we're able to use ctDNA as an early endpoint? To date, we were able to combine results from different assays using a percent change or a level of detection, but it's important moving forward to ensure that we have the accurate information about the assays to ensure that we have consistency across trials. For timing, our optimal timing for ctDNA collection is unknown. A lot of clinical trials use the ctDNA collection at the time when patients are coming in for treatment. That's more so for infused drugs. For drugs that are given orally, it might be when they have to come in for a resist measurement. And so what is the optimal timing and whether a confirmatory measure is needed? So we've assessed timing that has been included in clinical trials to date, and we're hoping that our analyses from Module 2 will help us to understand some of the dynamics of timing and help us to set the stage for how that information can inform future trial development. And finally, what's the definition of a molecular response? How do you define a responder? There might be different cut points in ctDNA metrics depending on the drug class or mechanism of action. So how do we ensure that we're defining molecular response appropriately? And then ultimately, how do we use that definition to show that one treatment group has a better response than another? And with that, we're hoping that our Module 2 analysis will help to answer some of these questions. But I want to shift gears a little bit and move into some of our diagnostic policy work focused on our diagnostics harmonization projects. Our diagnostic research projects ask the question, how do assays align, and can we find consensus to improve concordance? We disseminate a common set of samples with diagnostic or platform developers to evaluate variability in biomarker assessment. And our hope is that we'll promote alignment to support regulatory oversight of these novel technologies moving forward. We started this work back in 2017, with the Tumor Mutational Harmonization Project. TMB can identify patients who might benefit from immunotherapy. The project started in 2018. We have performed a few presentations. There are 2 manuscripts of the findings. The value of these outputs is we saw TMB-based FDA approval in 2020. And so having that approval and having the information that we found from the project can help to understand the value of using this test moving forward. It also informed discussions around diagnostic test oversight. The HRD harmonization project followed the TMB harmonization project, and HRD is used in ovarian cancer to identify patients for treatment with PARP inhibitors. We published a manuscript of the landscape assessment, and we presented the final data in February of 2024. And I'll share some of those findings with you today. And our next steps for this work is our digital pathology project with a digital pathology harmonization project focused on HER2 and breast cancer. We wrote a white paper that was a landscape assessment in this space, and we launched our project in February of 2024. We've also written some white papers and more of our policy side of things focused on diagnostics, and we've been involved in some of the conversations around VALID, the LDT rule, and the CDX pilot at OCE. So I just wanted to quickly touch on the TMB harmonization project. We shared a common data set of samples with 17 assay developers to assess concordance. And we were able to have an estimation, or we saw that the estimation of TMB varies across panels. So what I'm showing you here is compared to a common data set of whole exome sequencing, TMB, what was various labs, panel TMB that they called, and you can see there's some variation. We had we created some statistical calibration to achieve more consistent results, including a software tool to promote reproducibility and comparability across assays. And moving on to our HRD project, just for those who are not aware, homologous recombination deficiency is a complex biomarker that helps identify patients who might benefit most from a PARP inhibitor. We call it complex because different developers use different measurable indicators to create an HRD score. In our paper or manuscript that we published, we identified causes. So those are genes in the homologous recombination repair pathway that might be altered either by mutations or methylation. And then there's also consequences. So the result of having an altered HR pathway, things like genomic loss of heterozygosity, might be measured as a consequence of that pathway alteration. The patients are largely those with ovarian cancer, but we also see studies in pancreatic, breast, and prostate cancer. When we talk about benefit, it's improved recurrence-free survival or overall survival. And PARP inhibitors are a class of drugs that target DNA repair mechanisms. And so the challenges with this biomarker is that the complexity leads to different definitions of what constitutes HRD. We see that different assays have different cut points or thresholds, leading to inconsistency in how HRD is measured and interpreted. And there's variability in HRD measurements that may impact treatment decisions and ultimately patient outcomes. So for the HRD project, to address these challenges, we established this project, and which provides an opportunity to ask whether HRD assays results are consistent across different assays and what factors might contribute to any variability that we're seeing. As I mentioned, for phase 1, we published a landscape assessment in The Oncologist where we defined aspects of HRD and then also talked about some of the various characteristics that assays consider. For assay alignment, we provided HRD assay developers with 2 different data sets, an in silico data set and a clinical data set. Today, I'll be focusing on the clinical data set. And then finally, our clinical contextualization, just as important as finding these results is sharing them. So we presented at 2 different conferences. We had our Friends public meeting, and we have a publication that's forthcoming. For our study design, we distributed freshly extracted nucleic acids from 90 archival ovarian cancer samples. For our samples, we had 90 patients who had either stage 3 or stage 4 high-grade serous ovarian cancer. They were treatment naive and subsequently treated with platinum-based chemotherapy. The assay developers that we distributed these samples to independently sequenced the samples, then measured and reported HRD. We provided the assay developers with a survey to understand some of their assay characteristics. We had 17 different assay developers participate in this part of the project. All assessed BRCA1 and BRCA2 mutations. Those are 2 genes involved in the HR pathway and are commonly found in breast cancer and are commonly associated with HRD. And all of the assay developers assessed BRCA1 and BRCA2 to define HRD. And the cutoffs for HRD and range of values reported varied. So we saw a lot of different numbers. For example, one group looked at 0 to 100 for their range of values and used a cutoff of 42. Another group used that range, but then used 35. And finally, we had a group that used -20 to +20 and used 0 as their cutoff. So quite a bit of variety in how they're reporting their information. And then the distribution of the assay factors that were used to define HRD, GLOH, TAI, and LST are 3 different consequences. And so, about half of the groups were using those. And then mutations is referring to mutations in the HR pathway genes that are not BRCA1 and BRCA2. As I mentioned, everyone looked at BRCA1 and BRCA2. So this is other genes in those pathways. The NCI Biometric Research Program compared the results to determine the level of agreement. And the HRD harmonization working group reviewed and aligned on findings. For our working group, we met on a biweekly cadence for about 6 months to review the findings and align on the key outputs. A key aspect of our analysis is that we lack a gold standard for HRD, so we focused on an observed variability in the assays. So our first analysis was to assess concordance. And to do so, we calculated the % positive agreement, which is the percentage of samples that test positive by one test or assay, that are found positive by the second one. Here we're using assay A and assay B. We also calculated the negative percent positive agreement in a similar way, and then average positive percent agreement and average negative percent agreement, which were an average for each of the tests. In this case, we called HRD as positive and not HRD as negative. And so as an example, if we look across each row representing a different assay and each column representing a different patient, if we make all of these different comparisons and calculate a PPA for each of the different comparisons, we see the agreement analysis were performed across all possible combinations. And then we're able to create a median PPA for the overall data set. And because we had 17 different assays, we performed 272 comparisons for APA and ANA. And so here I'm showing you some graphs that demonstrate each dot represents a comparison for PPA, APA, NPA, and ANA, and then we found the median and interquartile ranges that are included at the bottom of each of the graphs. And so we see across all of the samples, we see a moderate level of concordance. The concordance being called moderate is kind of a qualifier that we agreed upon as a group, which doesn't tell much, but that's also because as a field, we don't really have the level that we would anticipate or expect, which is one of our major outputs from this project. We also assessed the impact of several clinical assay and sample characteristics on concordance. And here I'm showing you the results from our assessment of BRCA1 and BRCA2. And so we had the patients who had a mutated BRCA1 or BRCA2 on the left in the middle panel, and then the wild type BRCA1 and BRCA2 on the right. And we see that agreement is better for samples with mutated BRCA1 and BRCA2, so positive agreement compared to wild type BRCA1 and BRCA2, which we would expect. As I said earlier, BRCA1 and BRCA2 are involved in HR and drive HRD. So we looked at a variety of clinical sample and assay characteristics. CCNE1 amplification tends to be mutually exclusive from HRD. And we see that CCNE1 amplified samples have better agreement for not HRD. So the red dots are shifted up for the patients with CCNE1 amplification compared with the patients who have non-amplified CCNE1. When we looked at race and debulking status, we didn't see any associations. Similarly for the sample, when we looked at tumor purity, DNA quality, and age of the block, we did not see associations between concordance or agreement and those characteristics. It's important to note that in our study, we did have standards that we set for the tumor purity and DNA quality, which were likely higher than what we would anticipate in the overall field if it were a real world study. And then finally, looking at the use. So we asked the groups whether their assays were used as an RUO assay, research use only, or if they were used clinically in either clinical trials or for patients in clinical practice. And we didn't see differences between whether the assay, that was not a driver of agreement or that was not associated with agreement. And we also looked at the HRD cutoff. As I mentioned earlier, different groups use different cutoffs. And so we were interested to see whether there was agreement near the cutoff. And what I'm showing you here is two different assay results. We created an average concordance score. So averaging the concordance across all of the samples, all the assays for each individual sample. So each circle here is representative of a patient. The red line is representative of the individual assay's HRD score. Because we had differences in scores and our results are meant to stay blinded, we didn't actually put the values for what those scores are, but the red line represents where the cutoff would have been. And then we added a lowest smoothing curve, and you can see that the lowest point of the curve is near where the cutoff was. So we see less agreement near the HRD cutoff. And the next thing we wanted to do was to create a tile plot of our results to see whether there was, to be able to visualize the level of agreement and see if any patterns were emerging. So in the tile plot, the assays are rows and the clinical samples are included as columns. And so you can see each row here is represented by an assay and each column is represented by a patient. The dark blue represents HRD and the light blue represents not HRD. At the bottom of the graph, you'll have the BRCA status, so whether the patient had a BRCA1 or 2 mutation or if they had BRCA1 wild type, 1 or 2 wild type. And then percent HRD is taking for that individual patient the average of what the HRD call is. So if they had half of the groups called it HRD and half of the groups called it not HRD, it would be kind of more of a middle blue, whereas if all of the groups called it HRD, it would be a dark blue. We also, for each of the assays, we included information about whether the assay used various factors to determine HRD. And so on the right side of the table, you'll see GLOH, TI, and LST are all included as consequences. And then HRR is representative of the HRR genes in addition to BRCA1 and BRCA2. We clustered the samples and assays by relatedness, so we see how closely related each of them are, and it ultimately allows us to visualize some patterns that are emerging. As you'll note, we have assays J and K towards the top that only looked, if you see, they have a black bar for HRR, so they're only looking at HRR genes. And on the left side of the table, you'll see that there's a light blue area representing that those patients were not identifying, or those assays were not identifying patients who had HRD. And so we have a BRCA cluster. So those are patients who largely had BRCA1 or 2 mutations, and they were called HRD by the assays. We call this our consequences cluster because most of the BRCA1 and 2 most of the patients do not have a BRCA1 or 2 mutation. We also, as I was noting earlier, assay J and K only looked at HRR and they did not call any of those patients HRD, despite the fact that the majority of the other groups did. And then we have our not HRD cluster. So these are the ones where no one called the patients HRD for the most part. And so we took those clusters and we looked at survival analyses just using platinum treatment. Patients who have HRD often benefit from platinum therapy, and we looked at the 3 different clusters. And while we don't see any statistical differences, we do see that in the overall survival graphs that the consequences cluster and the BRCA cluster are overlapped. And trend towards improved overall survival over the not HRD cluster. Again, this is not statistically significant, but we're looking at trends to see that maybe this not HRD group, that this consequences group is part of the HRD cluster does not appear to be different from the BRCA cluster. And so our conclusions from this analysis, we saw a moderate level of agreement observed for the HRD calls across assays. Patient and sample characteristics do not account for the variability between assays. And so some recommendations for assay development, our hope is that we can identify the best approach for assays to report HRD to enhance consistency moving forward, align on expectations for analytical validation, and consider approaches for developing a gold standard, including the use of reference material moving forward. So these findings set the stage for harmonizing other emerging technologies. And in this case, we're moving into thinking about AI and ML. The next project we're tackling focuses on digital pathology. So I wanted to just give a quick overview of digital pathology. In traditional pathology, a trained pathologist reviews slides with a microscope. And in digital pathology, slides are scanned and computer algorithms assess the images using artificial intelligence and machine learning. A pathologist may review findings, however, whether the outputs from various models are similar or not is unknown. So for our digital pathology project, we first performed a landscape assessment where we characterized the potential of computational pathology in oncology drug development. We identified the challenges and solutions and platform validation and standardization, and we highlighted the need for an alignment. And so for this, we assembled a multi-stakeholder working group to develop a white paper and provide that landscape assessment of challenges and opportunities. We have since moved on to our demonstration project to assess concordance of biomarker measurements across different models, identify opportunities for alignment, and propose best practices. And here we're developing a demonstration project to identify variability among algorithms in HER2 scoring in breast cancer and support opportunities for harmonization. And so for our approach, we decided to look at HER2 in breast cancer for a few different reasons. We know that it's routinely assessed in breast cancer, so the likelihood of being able to identify a data set would be higher. It's a relevant drug target. There's ADCs that are emerging for HER2 that identify a new class called, or a new set of patients that are HER2 low. And there's multiple digital pathology tools that are used to measure this. And so we know that there's multiple groups that we can work with. So for this project, we have a digitized common set of digitized slides with relevant data. We have distributed those data to various platform developers who are running algorithms to run their models on the shared whole slide images. And we will next work on analyzing the results, so comparing the measurements across the models to understand the level of variability. We also have pathologist reads of these slides, and we'll be including that in our analysis. And so the objectives for this analysis, did you lose me again? Sorry, one more slide.
We did, but
Let's
I think you
see.
recognized that.
Yeah, got it this time. Okay.
Yep.
So
all right, we see it.
Objectives, our primary objective is to describe and assess the concordance of IHC-based HER2 scoring based on ASCO-CAP categories 0, 1 plus 2 plus 3 plus across different models. Our secondary objectives are based on these categories, assess the association of the level of agreement with characteristics of patients, specimen, and models, assessing the level of agreement on scores between models and pathologists, and then assessing the level of agreement between models that report percentage of cells exhibiting the staining patterns associated with each of the IHC-based ASCO-CAP categories. We'll also have a secondary objective to look at scores beside ASCO-CAP categories, so HER2 low, for example, if groups are able to provide that information. And so our projects largely are focused on bridging science and policy through collaboration. Our research partnerships represent unique opportunities to advance science and policy to bring innovative medicines to patients quickly and safely. And these multi-sector collaborations provide an avenue to rapidly develop new solutions, maximize resource efficiency, and advance science in a way that benefits all stakeholders. And as always, we want to say a major thank you to all of our project partners for participating in these various projects. And thank you to you for having me here today and for sitting through even with my technical difficulties. I really appreciate it.
Dr. Andrews, that was an incredible presentation, an incredible body of work. Colleagues, I'd like to open the floor for questions and comments. Dr. Andrews, I'll start out. So clearly, assay harmonization is a critical issue. And what do you think are the major challenges towards developing gold standards to benchmark diagnostic biomarker assays? You think that's something that the FDA could take on, or NIST?
I think it's a really great question. I think we have a few barriers we kind of have to overcome and agree upon before we get to that point. So the first is just sample availability. I talked more about our diagnostic projects in the sense of solid tumors, but we know, maybe it's a little bit more obvious in the liquid biopsy space. We can't ensanguinate patients in order to get enough, tissue to be able to understand the variability. And so what are the alternatives and what level of how do we feel as a community about the level of agreement using different reference materials, whether that's an in silico data set, which I think holds a lot of promise, whether that's something that's a contrived data set, but making sure that we have not only data sets that are biologically and clinically relevant, but that also can be used by multiple groups, I think is going to be really important. And as for who is the person, I think we're all kind of just waiting for somebody to kind of step up and take that on. I don't know whether FDA would be the right group for that, or who it would ultimately be, but I think it really is important for us to move towards that.
Indeed. Dr. Karlovich, you have a question.
Yeah, thanks, Nate. Hillary, great talk. Just wanted to say it's been great collaborating with you. I know we've been working together on the H.D. harmonization project. We worked on the TMB harmonization project before that. You've done a stellar job with all of this. I wanted to go back to the CT Monitor project and get your thoughts about a recent publication. So you presented some data, I think very compelling data. It's consistent with the literature, but you know, the magnitude of the retrospective analysis that you've done is unprecedented, right? You presented 8 studies in non-small cell lung cancer and with TKI therapy, and it's very clear from that work that ctDNA changes in ctDNA levels predict overall survival, I think, as I understood it, right? So that's a very important finding, looking at molecular response. Now, I don't know if you're aware of this, but there was a very recent publication in JCO Precision Oncology, and there was a study described of cediranib and olaparib. It was an NCI ETCTN study, and we often support these studies, by the way, but we didn't actually do the ctDNA analysis. But you're smiling, so maybe you're aware of this, but in that study, this was a heavily pretreated patient population. They had very high levels of ctDNA in their blood at baseline, and the response rate was rather poor. It was in the 10 to 15% range. So it was a different patient population, I think, than typically you see in non-small cell lung cancer in the trials that you presented, where you know, TKI therapy works very well in that population, right? You might get 40, 50% response rate. This population was a little bit different, and the findings were different. The gist of the findings were that ctDNA was not predictive of response in that patient population. I was wondering if you had any thoughts about why it wasn't predictive there, and is this something that we should be concerned about, and putting it in the context of your findings?
If I'm thinking of the same paper, was it different cancer types that were included?
Tumors.
Yeah,
So
all
there
solid
was a
tumors, but different
mishmash of
solid
cancer
tumors,
types.
right?
Yeah, probably the same one.
I think that was part of it for me. I think is the sample size was kind of on a little bit on the smaller side. So that's one thing that we need to think about is kind of overall effect. If we look at data set sizes, we're looking at really large ones. And so I think we've seen it in smaller data sets. And to your point, we've seen it in lung again and again. But I think we're also starting to learn that there's differences in different cancer types. We showed today, we might be seeing differences in different treatment modalities. And so I think the challenge for us is to identify the similarities and where those opportunities exist that we can use ctDNA. And then also identifying places where we can't is just as important, right? If we think about the resist space, we know that there's also, situations where resist is not the best. Things like IO therapy. We've created a whole new resist analysis because we know that pseudoprogression occurs. We know that patients who have bone-only disease often have some challenges with having reads there. And so I think, to me, it's not overly alarming, or it doesn't set us back. I think it's really critical to understand where the most important place to be using ctDNA is and to drive towards establishing standards in that specific space, which includes excluding those who it might not work as effectively for.
Yeah, thanks for that. And non-small cell lung cancer would be probably at the very top of the list of where it's probably going to work really well, right?
Agreed. Yeah.
Yeah.
And I think the thing that's really critical to remember about our work too is that we're looking at advanced disease here. And so another thing that we are really interested in, and we just haven't really had the studies to kind of pull the information together, is more in that adjuvant space or the neoadjuvant, depending on how we're kind of moving with lung cancer treatments, are there opportunities for understanding dynamics within an earlier stage disease? I think are just as important. It's kind of, we know pretty well in the advanced non-small cell lung cancer. So I think the more data, the better in this kind of thing, and making sure that we're being as transparent as possible is probably the biggest takeaway from all of our work, making sure when it comes to assays that we have the information we need to be able to make decisions and understand, limits of detection and assay characteristics, like whether it's, a tumor informed or it's a PBMC cleared, or if it's just an algorithm based where you remove chip that way. All of that information is really critical as we move forward. I don't think we're going to live in a world where you just say, oh, I looked at ctDNA. Having that context is going to matter.
Thank you.
Any other questions for Dr. Andrews?
Well, and if you think of anything after, please feel free to reach out. My email is handrews@focr.org. Be glad to talk further and answer any questions you might have at that point too.
Thank you very much for this outstanding high fidelity seminar. And I want to thank all of our colleagues for joining us here today. And I hope you all have a wonderful day.
Thank you so much for having me. It was a real pleasure. Appreciate it.
Thank you.